Hello students, we are here with our next topic, microfilament. As I have already discussed with you about cytoskeletal structures and different types of cytoskeletal elements, and I have already discussed with you about microtubules, its polymerization, depolymerization, structures, functions, and other features. And today we will discuss in detail about next type of cytoskeletal element, that is microfilament. Microfilaments are made up of actin proteins, and that's why they are also called as actin filaments. They have an average diameter around 7 nanometer, and these actin filaments are made up of a type of monomeric protein that is called as G-actin. This G-actin, it is basically a globular protein, and these G-actin proteins, they are highly conserved cytoskeletal proteins. They are highly conserved cytoskeletal proteins in case of eukaryotes. And these monomeric units of microfilaments, which are also called as globular or G-actin proteins, they are contractile monomeric subunits, or when they polymerize to form filamentous structures, these structures are contractile filaments. These G-actin proteins, they have molecular weight around 43 kilodalton, and they have associated ATP binding sites. And in the presence of ATP molecules and magnesium ions, these G-actin monomeric proteins are used to form a filamentous or a polymer structure that is called as F-actin polymer. It is fibrous one. F stands for here fibrous. And there are around 13 G-actin globular proteins which are used to form a single F-actin polymer, this fibrous actin polymer. And as I have written here, this is first G-actin, second, third, and so on. This is 13. Now these F-actin polymer, they intercoil around each other in a parallel way to form a double helical structures, and that helical structure, it is right-handed helix. Important thing to note down here is these F-actin polymers, they are parallelly aligned and they are intercoiled to form a right-handed double helical structure, where their plus and minus, plus that is fast growing end and minus that is slow growing ends are observed. And this F-actin polymer, when intercoil with other F-actin polymer, they form a F-actin filament. And these F-actin filaments, they are having number of ATP binding sites, where ATP and ADP, they can bind and they can carry out number of cellular activities. And in one of the case, these active sites, ATP or ADP sites, they are basically the active sites on F-actin filaments. These sites are used to form cross bridges with myosin heads of thick filaments during muscle contraction. Similar to that of microtubules, these F-actin filaments or microfilaments, they have slow growing and fast growing ends, where parallel alignment, it is achieved and these ends are observed. This is similar to that of microtubules, where alpha and beta subunits or heterodimers of tubulin subunits, they are used to form the polarity in case of protofilaments and subsequently in the microtubules. But here these are F-actin polymers which form a dimer that is having same polarity. And these microfilaments, they have this property of polymerization and depolymerization that is also observed at these two ends. And these, their monomeric components, they can be disassembled, they can be assembled, and this assembly and disassembly of these actin filaments, it lead to movement of these G actin proteins to the new sites or locations within the cells, and they can also reform or they can also repolymerize as when they are needed in the cell for specific cellular activity. And these, they form solid structures as compared to microtubules where a central hollow cavity is observed, but here they form solid rod-like structures. But these rod-like structures, they are thinner, they are more flexible, and they are usually shorter than microtubules. Actin filaments are associated with a type of motor protein, that is myosin protein, and with other motor proteins such as kinesin, dyneins, which are associated with microtubules. These myosins, they are also motor proteins, but they are associated with actin filaments, and they helps in contraction of muscles in case of animals, and they are motor proteins associated with animals actin filaments. There are different types of functions or roles which are carried out by these actin filaments or microfilaments within the cell, and first such type of role is cytoplasmic streaming. Cytoplasmic streaming helps in cellular motility. And second type of function of these microfilaments or actin filaments is formation of contractile ring, which is usually a type of structure formed during cell division in between the middle of the cell, which collectively forms polymers of, suppose these are globular G actin, and during cytokinesis a cleavage furrow is formed where the polymers of these actin filaments, these are F actin, they are formed, and subsequently the first phase of cytokinesis, this is cytokinesis second phase 1V, and here two cells are formed. So this cleavage furrow, it is cleavage furrow, and it is formed by these actin filaments, and this structure, this arrangement, it is called as contractile ring, and it ultimately helps in cytokinesis. And third role of these microfilaments, they helps in muscle contraction along with a motor protein, that is myosin. This is a motor protein, and actin protein, they forms filament along with three different proteins which are tropomyosin, troponin, and F actin. And these collectively two proteins, they form cross bridge and this cross bridge, it is associated with hydrolysis of ATP molecules, and that ultimately brings about muscle contraction. And there are three different types of troponin: troponin I, troponin C, troponin T. I will explain these different types of actin filaments and its composition in my subsequent lectures. But for here, with a brief restricting to the roles of microfilaments with respect to actin and its association with motor proteins, it is muscle contraction. So they ultimately helps in muscle contraction. And these microfilaments, they are also involved with another property that is called as membrane undulations, which are more common in case of protozoans, certain protozoans. And it is also a type of cytoplasmic streaming, and it is found in protozoans. Apart from this, these actin filaments, they are also influenced by a number of chemical agents which interferes with their polymerization and depolymerization phenomena. And these drugs, they are of different origins, and they influence different phases of uh, filament dynamics. And these are as, and these such as cytogelatin D. There is another one, and it is called as latrunculin, and there is also phalloidine, cytogelatin D. It is most common type of drug that is used for studying different phases of cytoplasmic streaming events, and these basically binds with plus end of actin filaments, and they prevent elongation, On the other hand, latrunculin, it binds with G actin in its globular phase, and prevent polymerization. Third type of drug, phalloidine, it binds with sides of actin filament, and interferes with depolymerization. So first one, cytogelatin D, it binds with plus end of actin filament where polymerization process has been initiated, and it further prolong or prevent its elongation. Latrunculin, it binds with globular G proteins and prevent their polymerization into actin filaments. And phalloidine, it binds with sides of actin filaments, and it interferes with the depolymerization phenomena of F actin into G actin. So this is all about microfilaments, and I will explain about intermediate filaments in my next lecture. If you like my video, you can share it, comment on it, and can subscribe my YouTube channel for many more such videos. Thank you.